come here just for the conference, but I have been in Europe for a few weeks, so over the jet lag, luckily. So very happy to be here today. I'm going to talk about my work studying the evolution of B1 and B2 Metallo-β-Lactamases. I'm going to go through the background fairly quickly so that I can touch on all our key results. So beta-lactam antibiotics are among the most commonly prescribed drugs, and they inhibit bacterial cell wall biosynthesis. And in response to that, bacteria have evolved Metallo-β-Lactamases, or MBLs, which can inactivate these antibiotics. And of course, they're a major threat to global health through antimicrobial resistance. So B1 and B2 MBLs have evolved from a much larger superfamily, which is structurally conserved, but has extensive sequence divergence. And we're interested in characterizing the evolutionary trajectories along which this beta-lactamase activity has emerged. And we want to use ancestral sequence reconstruction to do this. And for those unfamiliar, ancestral sequence reconstruction involves the inference and potential characterization of proteins at ancestral nodes in a phylogenetic tree. The challenge with B1 and B2 MBLs, and this challenge is not unique to this family, is that even within comparisons to its closest related subfamilies, there's still extensive sequence divergence between these. And we don't really have access to those ancestral trajectories until we resolve some of the more deeper relationships between these subfamilies. Now, this talk isn't really about this, but part of my PhD has been tackling this problem more generally through the development of a framework which iteratively expands the scope of the phylogenetic space of which you're interested in and continues to, so that you can continue to have confidence in larger and larger trees. But I'm not going to go through the details just because I want to touch on most of the key results that we've come to with the B1 and B2 MBLs. So we applied this framework to a starting tree of B1 and B2 MBLs and arrived at some convergence on the clusters that we were curating and a broadly stable and robust phylogenetic tree. And then we used GRASP, a tool that was developed in our group, to reconstruct the ancestral sequences in this tree. And then selected 4 along key trajectories and characterized them for their activity against 6 different beta-lactam antibiotics, spanning the major groups of the beta-lactams. And I know that was quite quick. Hopefully you're still with me. I'm going to go through the results of these characterizations of these ancestral sequences, but in the context of the key changes that are inferred to happen along these trajectories from our ancestral reconstruction. So at the last common ancestor of B1 and B2 MBLs, we infer 2 major events to have happened at the same time. One of these is quite unsurprising to us. We know that B1 and B2 MBLs have a dramatic active site rearrangement relative to the vast majority of the rest of the superfamily. The details here are unimportant, but there are 2 key changes that essentially completely change the metal binding geometry of the active site. The second was a little more interesting. We infer that the B1 and B2 MBLs evolved from a portion of the MBL fold superfamily space that have this extra C-terminal content, which you can see here. Most of these proteins, or all of these proteins, are essentially unannotated, uncharacterized, and we're unsure about the role of this particular extra content, but it's inferred to be lost at exactly the same time as the active site rearrangement. So we think that there's some synergistic role between the loss of this content, the active site rearrangement, and the emergence of beta-lactamase activity more generally. We also know that the B2 MBLs underwent some further active site reorganization, and this distinction between B1 and B2 MBLs is well known. Essentially, one of the key residues is lost or substituted, and the B2s become a monometallic enzyme. So characterizing the last common ancestor of the B2 MBLs, we saw very largely what we expected, largely selective activity against one particular class of beta-lactams, which is completely consistent with the activity of the extant enzymes from that class. So not overly surprising, but it was consistent with what we expected to see, and it gave us some confidence about the robustness of this reconstruction. Unfortunately, we had some trouble purifying the last common ancestor of all the B1 and B2 MBLs, and that work is still ongoing. But one point is that we, with impure enzyme, can only detect turnover on carbapenems, which is the same group that we selectively see activity against with the B2s. And it's always been, well, at least from our perspective, believed that this trajectory along here is where this specificity involved, but potentially this hints at ancestral preference towards carbapenems. Now, we thought that we may not be seeing the full spectrum of this activity because we may not have a very high concentration of enzyme. But then when we looked at the next node along this trajectory, we saw that this is the last common ancestor of B1 MBLs, I should say. These also seem to have a strong carbapenemase preference. Now, there was one of the cephalosporins that had weaker activity, but really meropenem, one of the carbapenems, is the only one that it showed any strong activity against, which maybe gives some further support to this hypothesis of ancestral carbapenemase preference. And then finally, there's a deletion that's inferred to occur along this very short branch that separates this small group of the B1 MBLs from the vast majority of the B1 MBLs. And that that deletion there is known to have occurred because it's a common feature of B2 MBLs. And while we don't have a lot of mechanistic understanding about what it does, it has been inferred that it may play a role in the substrate specificity of B2s towards carbapenems. And then when that was deleted, and I should note that these two sequences are very similar aside from this deletion, we see an opening up of the substrate profile. It becomes quite promiscuous on a bunch of different beta-lactams. We've also tested it on a few others that I haven't shown here, and it is very active. And we therefore believe that, yes, it does support, these findings do support a role for this particular extension here in the specificity towards B2 MBLs, but we think that it's probably actually blocking access to the active site for the other two classes of MBLs, of beta-lactams, sorry, rather than promoting carbapenemase activity more specifically. So I'm going to rush through the summary because I'm running out of time, but essentially we systematically curated this space and annotated some key events in the evolution of B1 and B2 Metallo-β-Lactamases, and in the context of that, characterized some ancestral sequences to reveal some new hypotheses about the evolution of their activity. We have picked the low-hanging fruit, essentially. We've looked at the really stark changes along these branches, but there are a lot of more subtle ones that we're now mapping onto the structures to see what their potential impacts might be, and working with our collaborators to give some insights potentially on how they could be used to target with inhibitor design. We want to look at characterizing some deeper nodes in this phylogeny. One, to look at the substrate profile of the ancestors, and potentially two, we may have found what we believe is a novel active site configuration, which I haven't had time to discuss here. And then we also want to look more narrower or shallower in this phylogeny, because in this small subclade, there is a high concentration of some of the most concerning B1 MBLs. So doing reconstruction, an ancestral sequence reconstruction in this space would answer some different questions, but related questions about the evolution of their function. And with that, I want to thank everyone involved on this project and our funders, the Australian NHMRC. Thank you.
Time for one or two questions while Paul sets up the slides.
Thank you for the really nice talk. I was wondering about with your ancestral sequence reconstruction. So my understanding of it is that at these reconstructed sequences, you have uncertainty about what the nucleotide or amino acid is at that sequence. So I wondered about how you came to a consensus of the sequence that you then used for your downstream analysis of what was happening with active sites.
Yeah, so when you do ancestral sequence reconstruction, you can do joint or marginal reconstruction. So a marginal reconstruction, you can target a single node and get a set of probabilities about the most likely residue in each position, essentially. What we did was a joint reconstruction, which gives you a the most likely assignment to all nodes in the tree. Because we wanted to have sort of some consistency between the nodes that we reconstruct. And in that manner, you don't get a quantitative likelihood, essentially. But it's a trade-off we sort of made to have that consistency. So we have then gone and checked the likelihood of those under a marginal reconstruction, those particular sequences that we used. And in three cases, it is very similar to the most likely sequence. The one that we have had trouble working with, there are some variations in some sites, and there's potentially that uncertainty may be contributing to the fact it's not an overly stable enzyme. But yeah.
Thank you so much.
Thank you.